questions. I know there's some technical terms in here, but for the most part, I'll try and be as simple as possible. And everyone can hear me okay? Yeah? So today I'm going to talk to you about a sponge that not only lives by itself, it lives with different sponges. And throughout my PhD, we wanted to figure out why the sponge is associating with all these different sponges, right? But before we start talking about sponge associations and why sponges, I think I pressed the wrong button. Sorry, yeah, there we go. It's important to know what sponges are. A lot of people don't know what sponges are. They're overlooked, like Mahelani mentioned. Sponges have been around for many years. They've survived mass extinction events, so over 500 million years they've been existing, have undergone through all the stresses you can imagine and still survived. We know that there are about 8,500 species of sponges that have been characterized, but there's an estimated 7,000 that remain to be described. So we have still a lot of work to do to classify sponges. There's very little efforts in this field right now. We're trying to get all these sponges identified and give them a name. And they make very important components of coral reef habitats. Not here in Hawaii particularly, but in the Caribbean. They make up to 80% of the biomass of the coral reef ecosystem. And with the associated microbial communities that they have inside their tissues, they can play very important roles for the reef environment. So this is what a sponge looks like on the right. It has tiny little spicules, which I'm going to go into throughout my talk. Spicules are the equivalent of the skeleton that we have to hold our body up. These spicules are made out of glass. They have tiny little cells that are flagellated cells, and they pump water through the sponge tissue. And a sponge is basically a community of bacteria that's associated with the sponge body. So I've mentioned that with the sponges that make up 80% of the community in Caribbean coral reefs and the bacteria that they are associated with, they are able to adapt to the nutrient-limiting conditions of the reef environment. So these reefs might be very biodiverse. You have sponges in many different colors, many different species, but the conditions that they live in are nutrient depleted. So they lack nitrogen, they lack phosphate. And scientists wonder, and it's one of the big theories from Darwin, who went to the reef and said, wow, this is a very biodiverse habitat, but how is it existing in such nutrient depleted conditions? Well, sponges, one of the answers to that is that sponges harbor microbial communities that allow them to adapt to these different conditions. And they exploit different niches of food to exist in these nutrient limiting conditions. So just to give you an example of what I'm talking about, we have a sponge that's called Xestospongia species on the right that associates with a very simple microbial community, and a sponge on the left that's a giant barrel sponge that associates with a very complex microbial community. So what you're seeing in the picture on the right is a lot of sponge cells within the tissue of the sponge, but no bacteria. And on the picture, let me see how I work this. Yeah, picture on the right, you see sponge cells, but that most of the tissue is made out of these little tiny bacteria, right? And these are termed high microbial abundance sponges, so sponges that associate with a lot of bacteria. So a sponge that associates with a lot of bacteria and has access to cyanobacteria, for example, is able to fix nitrogen, fix carbon, and make food with just the light that's coming in that allows it to photosynthesize and make these carbon sources for the sponge to feed on. Now, a sponge that doesn't have these bacteria relies more on what's floating in the water column as a food source, right? So these are two different lifestyles of sponges to give you an example of how these sponges are adapting to such nutrient limiting condition. Some feed on the particularly floating organic carbon, and some are phototrophic sponges, which feed on the products that are metabolized by or produced by the bacteria. Now, this comes with a demand. When you pump water through this very dense tissue of bacteria, you need a lot of energy to do that, while you have a tissue that's devoid of this bacteria. the water that pumps through comes a lot easier than the water that comes through this very dense tissue. So sponges that have developed this, that lack these bacterial symbionts will move water through their tissue a lot more easier. Sorry. So there are a lot of examples of how these food sources are made, and there are many scientists that have proven that the bacterial communities associated to this one sponge, which is the sponge that is most dominant in the Caribbean. You can find the sponge anywhere in the Caribbean, and it's the most common sponge of the Caribbean. Associates with cyanobacteria and can translocate those products into the sponge cell. So the sponge is phototrophic. If you shade the sponge, it will die because it relies so much on the sunlight so that the cyanobacteria can grow. And sponges also, with these microbial communities, sequester phosphate. And this is a recent study that got published a couple of years ago. What you're seeing in this diagram is phosphate that's around the water column, and the bacteria can store it in the form of these granules that fluoresce under microscopy, a very specific microscopy. And it's one of the reasons why we think the environment, the reef environment, lacks phosphate, at least in the Caribbean, where these granules were detected in very common sponges of the Caribbean. So we've mentioned different adaptations, lifestyles of how sponges adapt to the different reef environment in the photic zone of the reef. So these are areas that have a lot of light. When we dive down into the nooks and crannies of the reef, we have a different kind of adaptation and a different way that sponges are metabolizing and using food source. And this is the case, the interesting case of the sponge Halisarca cerurea. And this sponge is the size of the palm of my hand, so it doesn't grow very much, right? And the reason why it doesn't grow very much is because the food that it makes and the cells that it's making through the metabolism, through the metabolic processes, it actually sheds its cells into the marine environment. So it takes the dissolved organic matter that's around the reef environment, puts it into sponge cells, and just poops it out. So they don't grow very much because, yes, question back there.
What's it mean? DOM. Take it up and make sponge.
I'm sorry.
DOM.
Oh, DOM, dissolved organic matter. Dissolved organic matter. It's a form of carbon that is not assimilated by organisms on the reef. And I'm sorry, I should have explained that better. But it takes dissolved organic matter and they convert it into particulate organic matter, and scientists refer to that particulate organic matter as sponge poop. And that's very important because you're converting a form of carbon that is inaccessible to the reef environment, and you're making it into a form that is accessible to the reef environment. It can feed smaller organisms that feed larger organisms and contributes to the entire biodiversity of the reef. So this is a very crucial process, and scientists, when they discovered this, they were like, wow, sponges could be the reason why coral reefs are so productive, because they are able to take up this dissolved organic matter and convert it into a very simple form of carbon that other organisms on the reef can use. And it's a very complicated process, but I'm not going to get into it. So I've talked to you about how sponges adapt to the reef environment by the ways of their association with microbial communities. We call that a bottom-up process. And it's very important to not only consider the bottom-up processes that influence the abundance of sponges, but also the top-down processes. And the top-down processes are what predators cause on other organisms on the reef. So in Caribbean coral reefs, we have sponges that, these sponges are sitting around, they're not moving, right? So they need a way of how to either defend themselves or outgrow themselves out of predation. And that's what we see in Caribbean reefs. We have sponges that are chemically defended. They produce chemicals. You have a question?
Tell me a little bit more about that simple form of carbon in the poop.
The poop, the sponge poop.
What is it?
Okay, so I'll get into the cycle.
Small organic compounds?
Yeah, these are dissolved, dissolved forms of carbon that are not particulate and are unavailable biologically for a lot of the organisms on the reef. And carbon that's associated with, humic material or material that actually, this area of research is very interesting because a lot of people don't know what the dissolved organic matter is actually composed of. Yeah, no, and I can't tell you specifically what makes dissolved organic matter. I have a scientist back there in the back that might be able to. But anyway, fellow friends and colleagues. But the microbial, what's interesting is that the bacteria are able to do this at a rate of, it takes them 30 days to do this process. So if you see, the bacteria here are able to do this at a rate of 30 days to convert dissolved organic matter to particular organic matter, but it only takes the sponges 30 minutes to do this process. So it's very fast, very fast. And when it got discovered, the sponge community got very excited because corals take all the attention nowadays. And this is a very important process. Yes?
Can we go back to the birth of the sponge? The little sponge of the birth. How does that sponge pick up the bacteria?
That's a very good question. So when you have a baby sponge, right? So I'll go back to all these bacteria that are seen here. So a sponge, when it's born, sometimes it lacks a lot of the bacteria that's in them. Sometimes they're able to acquire bacteria from the adult. So the mama sponge will give it a bacterial package. Sometimes the larvae use this bacteria to swim around. So if it's a cyanobacteria, if it's a sponge that has cyanobacteria, it gives the larvae cyanobacteria. And with cyanobacteria, you produce glucose, right? And it's thanks to that cyanobacteria that the larvae is able to swim around and settle. So in a lot of cases, that's called vertical transmission of symbionts into the larvae. But not the entire microbial community is acquired that way. And sometimes the baby sponge is given what it needs to grow, but once it becomes an adult, it acquires bacteria from the surrounding seawater. Okay, so scientists are still fascinated by this fact, and they're trying to figure out how does a sponge select for its microbial community, right? So predation pressures. I was talking about how sponges need to produce chemicals to defend themselves from predation. And the scientist behind this is actually an advisor of my boss now. Well, he was doing his master's at the University of North Carolina, Joseph Pollack. And what he has done throughout the years is describe these chemical defense hypotheses that some sponges defend themselves by producing allelochemicals, and some resist depredation pressures by growing quickly, right? And not produce the chemicals. So it's very energy costly to make these secondary metabolites, these chemicals. And what you can see from this picture here, he took sponges that were palatable, sponges that were defended, and he made a little hole in these sponges, monitored the growth after 2 months in these sponges that were palatable and these sponges that were defended. And what he saw was that these defended sponges grew a lot slower than these palatable sponges. And this has ecosystem-based implications. And I was lucky enough to go around the Caribbean with fellow colleagues, and we published a paper saying that in areas of the Caribbean where you have areas that are protected, we can actually see that the distribution of sponges is affected by the predators that are around. So in areas that are protected, you're protecting the fish. There's a lot of fish that eat the sponges. It affects the distribution of the palatable sponges compared to the defended sponges. So in the areas in black are areas that have high predation pressure. And areas in white are areas that are protected from, the areas in black are overfished sites that remove the sponge predators. And areas that are highlighted in the white dots are areas where you have a lot of fish predators. So they're less overfished sites. And what we saw was that you have all these spongivorous fish, right? Something's not transferring very well, but let's go with it. In areas that are protected, you have the spongivorous fish that can take care of the palatable sponges. So you see the angelfish, the foulfish eating these palatable sponges. But in areas that you remove the fish that might eat these sponges, you have sponges like this Mycadian species that grows underneath corals in the Caribbean. And there's supposed to be a second picture on the right here where it says Mycada insatiable, where the spongivorous fishes are absent. You can see the sponge completely taking over the coral reef colony, right? So not only in the Caribbean do we need to be aware of the herbivores, right? So here in Hawaii, they tell you don't kill the herbivore fish because the macroalgae is going to overgrow the reef. In the Caribbean, we have the same problem, but the problem is that not only are we removing the herbivores, we're removing the spongivores, and not only is the algae overgrowing the reef, the sponges are also taking over the reef. So these palatable sponges grow very quick, right? So if we remove the predators that will control their growth, they will compete and outcompete corals. Does anyone have any question?
Yes.
Let me know. Yes.
We were recently in a tourist submarine off of Bonaire,
Oh, wonderful.
and we went down 131 feet.
Oh, that's awesome.
And we saw these tube-like structures, but they were very kind of greenish brown. They looked like this white palatable one here.
Okay.
Was that a sign of that, or are these just colored so we can see?
Sponges come in. So you're talking about like, was it more like this?
When they were tall, and they had a whole.
Yeah, it's something like this. This is not the Piscina caudiformis. Yeah, and that's probably what you saw because it's a very, very, very common sponge over there. That's yeah.
It was very tall.
Right.
Very tall. Thank you.
Right. Yeah, absolutely. Yeah, Bonaire has.
Oh, it's almost in Venezuela, it's in the southern Caribbean.
Yeah. All right. If there's any other questions, good. All right. So I've talked to you about how sponges, forms of sponges that have adapted a very specific lifestyle, right? So sponges that they're living by themselves and they're either palatable or chemically defended. The palatable sponges compensate for not producing the chemicals by growing quickly. The defended sponges don't grow quickly. Chemicals, I've talked to you about sponges that associate with a very rich microbial community and sponges that associate with a very simple microbial community. Do these sponges ever associate with one another to offset that restricted lifestyle, right? So will a sponge that has a high microbial abundant sponge, high microbial community, associate with a low microbial abundant community to offset that restricted demand of the lifestyle? So I talked about some limitations of having these strict lifestyles and how they deal with it. Are they associating with one another to offset these demands? And there are several examples in the literature that show how sponges grow together sometimes to offset space limitations. So if you go into a reef cave in the Adriatic Sea, where this was published, the first panel on the left here, a very famous sponge biologist named Rusler described that the sponges in reef cave habitats will grow with each other to offset space limitations. So it's a very crowded environment. And you'll find that sponges overgrow each other to offset that competition. And they apparently don't harm each other in the process. So they don't smother or completely kill the sponge that they're overgrowing. In the Caribbean, we have sponge assemblages that consist of where you have high predation pressures by, for example, the sponge-eating sea star on the right. You have sponges that get predated. And in this case, they're both orange sponges, but this one on the bottom, or this one, is a palatable sponge that just got predated here. You can see a little bit of white. Gets overgrown by this brown sponge, which is a chemically defended sponge. And it does that because it's a win-win for both sponges. So the chemically defended sponge uses the palatable sponge as substrate, while the palatable sponge on the bottom will get protected from predators like the sea star by having the chemically defended sponges grow on top. And there are more specific examples of this where you have two different species here of sponges, and this is a more specialized association where you have a sponge that is completely overgrown by this green sponge, this white sponge being grown by this green sponge for the same purpose of protecting itself from the sponge-eating sea star. In return, the sponge that's on top is higher on the reef and has access to particular organic matter and any particulates that it might need to feed on. So we go back to the big question of my research, and and it consists of, we found three sponge pairs, so three forms of sponges that live together, and the Xestospongia deweerdtae that I mentioned in the beginning of the slide is actually the pink sponge that's overgrowing the basal sponge, Black-ortis. And on the third, we have a third sponge pair, a close relative to this pink sponge that I won't talk about much because I don't think we're going to have a lot of time, but it basically belongs, it's a very close relative to this pink sponge.
If you
All right, thank you. So these were the dissertation topics of my thesis, but I'm going to go and identify the key components. We're trying to figure out why these sponges are associating. We didn't know what species they were, so I came across these sponge pairs, and we needed to describe them first to see what the heck they were, and try to determine what factors, whether the microbial communities were affecting the sponge association of the sponge pairs. Were the sponge predators affecting these sponge pairs? Were these sponges associating to protect themselves from the predators? And as a final chapter, we went into whether the ability of this sponge to grow in different ways could serve as an advantage to deal with the effects of a changing climate, so ocean acidification and temperature increments. So we go into the taxonomy of these sponges. I'm going to walk you through it. We dissected the epibiont, which is the Xestospongia deweerdtae, and this is the hitched lifestyle of the Xestospongia deweerdtae growing on top of this basibiont sponge. We dissected them, and taxonomists have to look at everything in order to describe a species. So we looked at the spicules that make the sponges, and what we found was something really interesting. We found that this sponge that grows on top of the basibiont is Xestospongia deweerdtae. We didn't know it at the time, but it produced these spicules that were very similar to the free-living form of this sponge. However, there was a big difference in the size of the spicule. So the size of the spicule of the free-living form was a lot bigger than the size of the spicule of the associated form. Yeah.
Spicules, silicates,
Yes.
carbonates, or protein?
Silicate is, it's made out of silica, so glass.
On all the sponges?
Yes, all these sponges. So these sponges make an incredible array of spicules that are made out of glass, and if you put them together, you can build a skeleton out of it. And this is how sponges stand up, basically. Right?
How often have you been impaled in playing with these?
I haven't been impaled because I use gloves. But at the beginning, when I was very young and stupid, yes, I had an itch on my fingers many times.
Did I get that right? All the spicules are made of glass?
Okay, not all spicules are made out of glass. Some spicules are made out of calcium carbonate as well.
No, I was thinking of some people in Florida, on the east coast of Florida.
Yeah.
They get sponges from prison. It's not going to be made of glass because they squeeze them.
Right. So those don't have spicules though.
Yeah.
Yeah, those are made out of collagen fibers, and those are bath sponges. Those are sponges that you can actually bathe with, and those don't have any spicules. Yeah, so to use. So we were very interested to see why the skeleton structure of this free-living form was bigger than the skeleton structure of this associated form. So we looked at several areas in the Caribbean. We went to Puerto Rico, we went to the Bahamas, and we went to Panama, and we surveyed the Caribbean for these two forms of the Xestospongia deweerdtae. to see if one was found more abundant than the other. And nowadays, we not only look at the morphology of the sponge, but we look at the DNA of the sponge to see if what we have is actually what we're saying we have. So a sponge that produces different kinds of spicules does not necessarily mean it's a different species. We need to look at the genetic code and look at these regions that are very specific to identify species at the genetic level, right? So we did that, and what we found that there were all these different forms of Xestospongia deweerdtae that have different morphologies, free-living, lifestyle associated, thickly encrusting, and these were all 99 to 100% DNA sequence similar to each other between all the different polymorphs. So this is the way that we were able to say, wow, this sponge, big spicules, small spicules, it grows with these chimney structures, all of those are the same species. We were able to say that with the DNA that we got and the sequences we compared, and these are all 99 to 100% sequence homologous. What the spicules look like, and we ask ourselves, okay, so we have the same species growing in different forms, large spicules that are small, but one consistent thing that we see is that between the associated form and the free-living form, there's a size difference regardless of the area that's found. So these are sponges that were collected in Panama, and in Panama we have a lot of nutrient output, specifically silica output, that affects the size of the spicule. So in areas that you have larger output of silica because of the rivers that are around, you're going to see that the sponge will produce larger spicules. And in areas like the Bahamas, nutrient input, very little silica input, and smaller spicules. But regardless of that phenomenon, regardless of whether we had higher silica or low silica environments, the phenomenon of seeing that were larger in the free-living form was consistent regardless of the silica. And we tested this, and we had significant outcomes that the associated forms had smaller spicules versus the free-living form on the right. And this was tested throughout the Caribbean. So we believe that this is a factor of the symbiosis. Maybe another sponge is receiving less nutrients to synthesize the spicules, or maybe by associating with this basidian sponge, the sponge doesn't need to produce spicules that big, right? So by producing spicules this big, and you're a free-living form, you might need something to create more structure because you're not associated with anything. Those are two avenues of possibility. So with the basidion, we did the same thing. We extracted the spicules, and we looked at the spicule composition just to describe it. And what we found was, when we look at the genetic barcode of the sponge, we found that it was a new species of a sponge that's called Placoertes. It's a different class of sponge, completely different class of sponge from the epibion. For the second sponge pair and the third sponge pair, we saw that the Placoertes that was found in the first sponge pair, this was a different species. So the basidion is a different species from the first basidion. Basidion can associate with two epibions, with the deweerdtae, but also this blue sponge. I don't know if you can see that. There's a little blue sponge growing on top of the basidion. Sponge pair 3 associates with two epibions. Geographic distribution of this sponge pair, Placoertes deweerdtae, and we try to find free-living forms of it, of the basidion sponge, right? Forms of the Xestospongia deweerdtae, but we haven't found free-living forms of this basidion sponge. So we believe that this is an obligate association for this basal sponge. We also find tiny little epibion growing on top of it. So I don't know if you can see that the surface of this brown sponge has the pink sponge growing on top of it. So this is an association that has developed from a very early life stage. So they're potentially time and forming these sponge pairs on the reef in a very early life stage. So for sponge pairs 2 and 3, the same thing. We can't find a free-living form of that basidion sponge. It's always obligate. Well, we believe it's an obligate symbiosis for this basidion sponge. Look at the overall distribution of the sponge pairs and the free-living form of Xestospongia deweerdtae, we see that there's a 4 to 23 times more abundance of these sponge pairs compared to the free-living form of Xestospongia deweerdtae. So that either means that the sponge pairs are getting eaten less, more of the free-living form, or that there's a higher reproductive output of the sponge pairs, and they're doing a lot better in, from a reproduction standpoint compared to the free-living form of Xestospongia deweerdtae. So we know what their species are. We can start asking ecological questions as to what is influencing them. And when we take a section of the sponge, right, of these sponge pairs, we can see that these sponges are intertwined one with one another, right? So we have the epibiome growing on the basibiome, but they're intertwined with one another. And we ask ourselves, well, one sponge clearly has a denser tissue than the other sponge, right? So the Ortiz has more of a denser tissue than this loose tissue of the epibiome that is growing through its tissue. And if we go back to the introduction where we said that sponges that have a high microbial abundance have a high microbial abundance of the bacteria are going to take a lot more effort to pump water through their tissue than sponges that don't have bacteria. These Xestospongia deweerdtae belong to a group of sponges that have a lot of bacteria, but also don't have a lot of bacteria. So our hypothesis is that the reason why these sponges are growing like this is potentially because the epibionts might be releasing that energy demand of the high microbial abundance sponge to pump water through its tissue. And in exchange, this epibiome has access to unique microbial species that it wouldn't if it wouldn't associate, right? So we're testing this hypothesis. That is our hypothesis. The sponges, one associates with a lot of bacteria, the other does not associate with a lot of bacteria. They're associating together to offset the restrictions of that lifestyle. So what we did was that we took a section of the sponge, we counted how many bacteria there were inside the sponge cells, in addition to looking at the DNA of the microbial communities that are associated with them. And we used a stable isotope analysis to see what they were eating and see if the signatures that we got from these elements were the same or different for both sponges, and see whether they were competing for the same nutrient source. When we look at the sponges under the microscope, we stain them with this dye that fixes itself to DNA, and we're able to see it under a very specific form of microscopy. What you're seeing here are cells that are sponge cells and bacterial cells. Sponge cells are those that are really, really large, and the bacterial cells are the tiny little dots. It's really hard to see from this image because of the contrast, but right off the bat, what you can tell is that on the right, you see epibionts that have highly nucleated sponge cells, and on the left, we have basibionts that not only have the highly nucleated sponge cells, but the bacteria that are associated with these basibionts, a lot more than the epibionts. So we counted many, many sections from each sponge pair. So basibionts are on the left, the epibionts are on the right of the 3 panels going from top to bottom. So when we count the sponge cells on panel A and count the bacterial cells on panel B and do the ratio of these cell counts, we see that the basibiont sponges have a lot of bacteria in comparison to the sponge cells, and that the epibionts, which, so these are the basibionts right here, these are the epibionts. You can see there's a clear difference in the number of bacteria and sponge cells that they have. And overall, these results strongly suggest that the basibionts have a lot more bacteria than the epibionts. And with that, we know that these basal sponges are potentially harboring a lot more bacteria than the epibionts are. We didn't detect any cyanobacteria in our study because they live in reef cave habitats. And I don't know how much of this I'm going to present because I don't know if we have a lot of time, but I'm going to try and move it forward. But basically, we did the same thing we did for the first part of our talk, where we extracted the DNA and we wanted to look at the at how the DNA of all the different bacterial groups, right? The classification of all the bacterial groups with the DNA are in both the epibiont and the bathybiont. So we have the 3 sponge pairs, including the free-living form of Xestospongia deweerdtae. And we're doing this to see what kind of bacteria we have in our community, right? So we extract DNA to do this, and we submitted samples to an initiative that is out there to characterize the entire Earth microbiome. So there's a pot of money separated for anyone who wants to take any sample from any organism to look at what microbiome is associated with that organism. So we took advantage of this initiative because it would give us really good data. We submitted a bunch of samples, and what we saw right off the bat was that there are different diversity indices on my x-axis. We have one type of measuring diversity, which is OTU richness. It measures alpha diversity, so all the bacterial species that are in your sample. And on my y-axis, I have an index that tells you how diverse your sample is and how the different species in your sample are distributed in your sample. Basically, what I want you to take from this is that the bathybions were a lot more diverse than the epibions in each of the different locations, Puerto Rico, Bahamas, Panama, Mexico, and that these epibions had a very low microbial diversity compared to those bathybions. This further supports the hypothesis that the bacterial communities of the bathybion are not only more abundant, but also more rich. And so when we look at how these communities cluster with one another, we have different colors on the right of the panel grouping together because they are from the same species. Different colors belong to different species, but so for example, in the red we have Xestospongia deweerdtae free living. In the pink we have Xestospongia deweerdtae associated, and that's what I really want you to look at here. The red and the pink dots from the 4 different locations. You see that they are grouping separate from one another. And the reason why they're grouping separate from one another is because the associated form of Xestospongia deweerdtae is more closely related. The bacterial communities are more closely assembled to the bathybiont sponge. So they're acquiring bacteria that are from the bathybiont sponge. So you can see that shift I'm pointing here with the arrows. And that the epibionts, Xestospongia deweerdtae, which are circled in pink here, the associated forms are more diverse than the free-living form in red. So this Xestospongia deweerdtae is acquiring more bacteria than the free-living form. So this is what the different bacterial groups look like in each of my samples for Xestospongia deweerdtae collected in Panama. And in other locations of the Caribbean, Puerto Rico, Bahamas, and Mexico, we have these different bacterial groups in different colors. And what I want you to see, the only thing I want you to see from this is that between the free living, the FL, and the associated forms in these different locations, the chloroflexia are a lot more abundant in these associated forms, and the acidobacteria are a lot more abundant than the free living forms. And that's because the associated is getting those bacteria from the bathybiont. So here we have epibionts on the left and the bathybiont on the right. We have that the chloroflexi make a very significant component of the community of these bathybiont sponges, and that they're potentially supplying these epibionts with this bacterial species that the free living form is unable to access because it's not associated with the bathybiont. So in summary, the bathybiont sponges are HMA sponges with a more complex microbial community, and that the epibionts are LMA sponges with a more simpler community. And we're observing a new form of symbiont transmission. So your first question earlier was about, wait a minute, how are the sponges acquiring bacteria? Well, I mentioned that the adult sponge gives the baby sponge bacteria through vertical transmission, and that the sponge can get bacteria by filtering water in the water column and getting bacteria that way. But this is the first time that we notice that the sponge can get bacteria from another sponge. in symbiosis. So it's a new form of horizontal transmission for sponges that's been observed. Okay. You have 3 minutes, and that's it. And then questions? Yeah.
I'm sorry, can I quickly ask?
Yes.
I did come in late, so I apologize.
No.
for talking about this. What is the advantage of having more bacteria?
If you have, that's a very good question. The advantage of having more bacteria is that you can have different pathways to acquire different forms of food. So bacteria carry out very important biological processes. Like, for example, if I mentioned in the beginning, if you're associated with cyanobacteria, you can photosynthesize carbon into glucose, and that glucose is a food source for your host. And corals do it with the algae, and so Xantelli. So if it's like an avenue for a new form of food that you can acquire. Not always is that the case, because there's a lot of microbial processes that occur, but sometimes it can lead to the availability of food that you would not have if you weren't associated with a specific microbial community. Yeah.
With regard to the bacteria, not the photosynthetic ones, but how many of the species of bacteria are similar to the ones that we have in our gut that help us live?
Right. So that's a very good question. So the question was, how are the microbial communities in sponges similar to the microbial communities that we see in our guts? Right? So that's a very good question, and I don't know if I can answer it correctly. But I can tell you that the diversity that you see in the sponge microbiome is unmatched. So in a survey that just took 80 sponge species from the world, just 80 sponge species, the diversity seen in these 80 sponge species is equivalent to the diversity of bacteria observed in the entire world's ocean water. Okay, so it's remarkable. We're talking about 40,000 species of bacteria that have been found in sponges recently in this Earth Microbiome Survey. In just 81 species of sponges. So we know so little about this diversity that we have still a lot to learn from it. But our microbial gut fauna, we know a little bit more of. I don't know if. I'm not sure how they relate to each other, but that's a very good question. Yeah.
Do you think that the food source even lower than this process that produces the energy or glucose that provides another animal for it?
Yeah. So is there a food source? So if there's a food source that if there's a food source that goes below glucose or any of the byproducts that bacteria make, right? Yeah, so dissolved organic matter that I mentioned earlier, right, is a food source. Bacteria and sponges are able to take up that dissolved organic matter and make it into food. So and sponges are able to do that. Sponges are able to make food out of things that other organisms can't feed on. So there is, and if, for example, these sponges are able to do that, are able to convert DOM, make food out of it, and I can associate with that, then you're at an advantage over other organisms. So that's a very good point.
So we as a human come in and we eat a fish in the middle range, but it's all dependent on what started off.
Exactly. So if it wasn't for, well, I'm not going to say all the sponges, but if it wasn't for these sponges, a lot of the food that's available for the reef might not even exist. And this is one of the reasons why you got to start feeding the smaller fish to feed the big fish, right? And this is where potentially it all starts in coral reef. Ingrid, you had a question.
I was going to say another fun fact that a lot of pharmaceuticals come from marine organisms, and about 30% of those pharmaceuticals are derived from bacteria that in found in sponges. So they're really, they do crazy stuff that makes them really interesting.
Yeah.
They're food for turtles.
They're food for turtles. So with this untapped source, back to Ingrid's comment, of them associating with bacteria, it's an incredible source of biotechnology, right? So bacteria, we derive so many biotechnological applications from bacteria. Amylase, we have different detergents that are produced by, well, we have different products that are produced by bacteria, and these sponges are an untapped source of this biodiversity that could produce several antibiotics and pharmaceuticals for future human applications. Yeah.
I saw a documentary this week actually about sponges, and in Florida they're doing research on sponges as something that they're producing in bacteria as a cure for pancreatic cancer.
Yeah, there you go. Yeah, so I'm actually, I should, now that I see where this is going, I should have given a talk more on that. I actually am a trained natural product chemist. So when I was in Puerto Rico, I dedicated my undergraduate years to taking a sponge, putting it in a blender, and purifying drugs. And the problem is that the FDA knocks out these drugs from their assays very quickly. It takes years for a compound to be purified to go into the market because of many technicalities. But yeah.
Can you go back to the first chart?
To the first?
Yeah, first chart.
The first chart at the very beginning. Which chart?
Number one.
Number one. Very first one.
How often do sponges
Some sponges have larvae all year round, actually. Yeah, some don't. Some only reproduce in the summertime or warmer months, periods. So that may vary. But
see it with the eye?
yes, you can cut a sponge up, open it up, and see baby sponges in it. It's pretty cool.
Okay, you don't have to, anyhow.
Yeah.
What is the Xestospongia filters? That was in the first.
Filters.
So the very first chart you had on.
Was it this one?
Very bottom.
Right here.
Sessile
Sessile filter feeders. Yeah,
What
okay.
is that?
Okay, sessile filter feeders is anything that's stuck in one place and filters water. Clams are sessile filter feeders. Sponges are sessile filter feeders. They don't move, and they just feed off the water that they are drawing into their system.
How do you spell that?
That might be wrong. No, that should be right. Yeah, sessile. S-E-S-S-I-L-E.
So Jan,
Yeah.
we have 10 minutes longer in this round, so why don't you take 5 minutes to kind of wrap up? what you
Wrap
wanna
up.
present?
Yeah.
We'll use 5 more minutes for.
Yeah. So let me try. Yeah.
Because I know you wanted to get through.
Well, yeah. I'm so sorry. No, it's okay. I'll try and talk very quickly because it's very cool. The next part was probably cooler than this part. It involves a natural product, and I was going to go into the chemistry of it. But basically, as an undergrad, we isolated these compounds that were antitubercular compounds from the sponge on the bottom. And what we found was that these compounds are translocated into the epibiome sponge for protection. So not only is the bacteria being exchanged, and we tested these compounds with fish, and I wanted to show you a nice little video. Sorry about that. But we actually detected these compounds. Yeah, the compounds that are produced by the sponge. Yeah, anyway, these are the compounds that I'm talking about, and they look like that up there. We detected them in the basal sponge has a lot of them, a lot of these compounds, and we detected a tiny amount in the epibiome of deweerdtae. And the reason why that's important is because when we tested the crude extracts on fish, right? So we fed fish extract of the sponge, they would not feed. So these are a number of pellets that the fish would eat, and we tested it on 2 fish species that are generalists. They didn't eat a single pellet out of several dozen pellets that we fed them from this basibiome sponge. And we diluted that crude extract about 250-fold, and it wasn't until we diluted it up to here the extract became palatable. So you need a very tiny amount of this compound to make the whole thing defended against fish predation. So the tough sponge is using that compound to protect itself from predators.
What about Hawaii sponges?
That's why I'm here. So maybe next year I can give a talk on Hawaiian sponges because I'll learn a little bit more about them. So I came here as an NSF postdoctoral fellow to laboratory of the sponges of Hawaii and use molecular tools with in the lab that I'm currently in to identify the species of Hawaii. And unfortunately in Hawaii we have a lot of species that have come from different places because of sponges that are in the ballast water of ships, and so they come as an introduced species. And a lot of the sponges that we see in Kaneohe Bay are believed to be introduced species, but we don't really know that. And we're trying to decipher whether that's something that is actually true. If the sponges that we have in Kaneohe Bay are locals from Kaneohe Bay or are they introduced? And so we're gonna try and figure that out through molecular techniques, and that's why I'm here in Hawaii now. So all that is work that I've done in the Caribbean. So I'm trying to use that expertise to try and answer those questions here in Hawaii.
A lot of sponges in Kaneohe Bay.
What's what?
There are a lot of sponges.
We have found about, right now, right off one location, we found between 40 and 60 species of sponges, and there could be a lot more than that.
Kaneohe.
Kaneohe, sorry. Yes.
So we know that the sponges and the bacteria are very important,
Yeah.
and so sunlight is important. Are there any species of sponges that shy away from sunlight?
Yes. There's a lot of sponges that are found in caves and very low light environments, including the species that I just talked to you about today, where sunlight is completely devoid. You have a very rich diversity of sponge species in reef cave habitats. Yeah.
Does any of the sponges end up in the cuisine of any people?
Not that I know of. Not that I know of. Not that I know. The only way they're used, like for commercial use, is to bait, and they have been used for thousands of years by the Greeks. Actually, a lot of people farm the sponges and they sell them to different communities, and they're shipped to Europe because they're a delicacy to bait with. Yeah, they bait. Yeah, so these sponges that you were mentioning, someone in the public was mentioning, that don't produce the silica specules and are very spongy. Those are very soft and very nice to bait with. So I think you can find them in some places, maybe Marshalls or something. Might sell them. Yeah.
Did you want to share the video?
Well, yeah, the video is not very eventful, but it shows you it shows you how we classify a sponge that is deterrent. Here, let me see if I can find it. Deterrent or palatable. So I've talked about chemically defended and palatable sponges, and this is a video that shows how you determine if a sponge is defended or palatable. And they do it by taking crude extract of the sponge and putting it into squid paste, right? So let me show you this slide right here real quick before. So we take squid paste, we take the sponge extract and put it into the squid paste. The way the assay works to see if what you have in your sponge is nasty or not is that you have a control pellet that has no chemical extract in it, and you have the treatment pellet that has the extract in it. Every time you feed the fish, you gotta give it a control pellet, a pellet that they will like without the food extract, so that you know your assay is working and that you have a fish that's cooperating. And then you give it the treatment pellet to see whether it rejects it or if it accepts it. And you can't run the assay if the fish is not in the control pellet that has no extract, because your data is gonna be flawed, right? So if the fish eats more than 6 pellets, and we're talking about a lot of replications using many different fish species, feed it 10 pellets. If it eats more than more than 6 pellets, it's a palatable sponge. If it eats less than 6 pellets, it's a chemically defended sponge. And this video that I'm about to show you was developed by a good friend of mine, Marika Marty, who actually has a link into how this works in more detail if you were interested in learning more about it. Where is it? Yeah, here we go. So yeah, here's accepting a pellet. This is what it looks like. You feed a fish, the fish will eat the pellet immediately, and for rejecting a pellet, you can see that nobody likes the pellet. Okay? So it will spit it out, and it will spit it out, and it will continue spitting it out until it falls to the ground. And we do this many, many, many times to get the data that we got, that you get here. So you get mean number of pellets eaten, all the sponge species of the Caribbean, and whether or not they're defended or palatable. So this is the basic study that was done to confirm the whole hypothesis of the impact of removing spongivorous fish in the Caribbean or not. So this is what gave us the data to say, hey, this sponge is a very toxic sponge and it's defended. Oh, this sponge is not defended. And this inspired a bunch of different studies, and yeah, a bunch of studies that led to that. But sponges are very complex. So my fellow colleague back in the back will probably say, you're wrong, because in Indonesia, predators don't matter. As long as there's enough sponge poop or poop in the water, it will grow better. The habitats are very different. And Kaneohe is a very peculiar place, and Kaneohe is a very peculiar. I am so sorry. It's a very complicated place, and from different perspectives, a fascinating place. So we're going to try out some of these theories